I'm the pharmacist in charge of the quality control laboratory within the MEARY Center. I have been trained in the field of biotherapies in Paris hospitals, and I have completed my training in the Clinical Investigation Center for Biotherapies at the Salpêtrière Hospital in Paris. I have more than 10 years in experience in the development and quality control of hematopoietic stem cells and advanced therapy medicinal products. I also hold a PhD in immunology, and I have a strong expertise in basic and translational research in cell and gene therapy. Our team in MEARY produces cell and gene therapies such as mesenchymal stromal cells, Treg lymphocytes, CAR T cells, NK cells, and more other ATMPs. All ATMPs produced in MEARY Center are for phase 1 and 2 clinical trials. Our job is to produce new cells and new gene therapy drugs to treat patients suffering from different diseases, such as autoimmune diseases, hematological diseases, cardiac diseases, and so much more. We are doing personalized medicine because in some cases we are treating the patient with its own cells. We take the cells of the patient, we modify the cells, and then we re-inject the cell to the same patient in order to cure his disease. For instance, we use autologous T cells to produce CAR T cell and then re-inject this cell to the patient to treat his leukemia. We use the Conical products in many of our manufacturing processes. For example, we use Conical products for manufacturing of mesenchymal stromal cells. We use these cells in 2 of our clinical trials. The first one was the STROMA-COV2 clinical trial coordinated by Professor Antoine Moncel at Salpêtrière Hospital in Paris. We use mesenchymal stem cells to treat COVID-19, acute respiratory distress syndrome. We also use mesenchymal stem cells to treat traumatic brain injury. This clinical trial was coordinated by Professor Vincent Degos. And in these 2 projects, we use Conical product to isolate and expand mesenchymal stromal cells. To produce mesenchymal stem cells, the first step is to isolate these cells from umbilical cord. So we isolated these cells using CellSTACK 1 layer, we expanded these cells, and then we harvested these cells using CellSTACK filling accessories, and then we centrifuge these cells using Falcon or 500 ml corning tubes. After this step of harvesting, we re-expand the cells using CellSTACK 10 layers. So we have done an amplification of the cells using HYPERStack 12 to 36 layers, and then we harvested the cells. We are looking for an improvement of our manufacturing processes in order to lower the cost of the ATMP production. We need to find at the right cost the tool for small batches, for example for autologous ATMPs, but we also need to find the right tools for huge batches such as we need in allogeneic ATMPs. We need also to find tools for closed manufacturing system, and we need to have GMP compliant medium for different cell types at the right price, including the medium and also the cytokines.